Chapter 4 is looking at a typical animal cell and a typical plant cell, and looking at the organelles that we find within that typical plant and animal cell. So you should have out your outline for Chapter 4 and, of course, your textbook as we go through Chapter 4 here. And you notice at the beginning of the outline for Chapter 4, it says eukaryotic cells. So this is a term that we learned at the beginning of the semester already, but it's really important to point out here for Chapter 4 because this chapter only applies to eukaryotic cells. So any organism that's classified into domain Eukarya. It does not apply to bacteria nor domain Archaea, because neither of them have organelles. So this material in Chapter 4 only applies to eukaryotic cells, which have a defined nucleus, and that's the definition of eukaryotic or eukaryote. The next thing in the outline is plasma membrane. So I mention it here because it's an important structure within a cell, and the organelles themselves are made of plasma membrane. So plasma membrane is really important for living cells. Notice I have there, as far as a definition, a phospholipid bilayer in which protein molecules are embedded. So those phospholipids I mentioned to you in Chapter 3. Then I had said there that when we get to Chapter 4, we would be talking about phospholipids much more, as well as then Chapter 5 especially. Chapter 5 is where we actually learn about plasma membrane itself. So we mention it here, but then Chapter 5 is really where we'll spend a lot more time with phospholipids. So notice bilayer meaning two, so a plasma membrane has two layers of phospholipids that are sort of sandwiched together, and then there's protein molecules embedded in it. And I put a brief picture, a small picture there in your outline book or in your outline for Chapter 4, and you can kind of see those things labeled there. So we'll go through that in more detail in Chapter 5. So I guess let me say it this way, that I'm not spending a lot of time in Chapter 4 on plasma membrane, even though I'm saying to you how important they are, because they're actually so important that they get half of their own chapter in Chapter 5. So there's no need to spend part of Chapter 4 talking about plasma membrane when literally half of Chapter 5 is devoted to that, because that's how important they are. All right, so moving on, cytoplasm. So cytoplasm is the fluid that sort of bathes the organelles, or you could say that the organelles are suspended in the cytoplasm. I guess you could say it either way, and it would be true. So cytoplasm is really a water-based medium with salts and organic molecules in it, like enzymes. Then you see the term organelle. So organelles are in plants and animals. We also find them in fungus and protozoan. So any eukaryotes have organelles. And they are plasma membrane bound structures that have a specific function to them. So really the point of Chapter 4 is to go through what are the typical organelles you would find in an animal cell, which is what you're looking at here on the screen. If I go to the next slide, this is a typical plant cell. So what we're going to do, literally the whole chapter is set up that way. It's a survey chapter of the organelles that you would find in a typical plant and animal cell. So I'll come back to these two pictures here occasionally as we go through and just kind of point out similarities or dissimilarities. But with that sort of brief introduction, let's go ahead to the second page of Chapter 4 outline, and it's underneath the plant cell there where it says nucleus. So the nucleus, and that's what you see on my screen here. So the nucleus is the first organelle we're going to talk about. The nucleus is a double plasma membrane structure. So it's got two plasma membranes, and we call that, and you can see in your outline, the term nuclear envelope. So the two plasma membranes sort of sandwiched together here, we call that the nuclear envelope. So it is the outer boundary of the nucleus. The nuclear envelope has holes in it, and here's that word pore, P-O-R-E. So the nuclear pores are these little holes that you see all over and throughout the plasma membranes that make up the nuclear envelope. Those nuclear pores, those holes in the nuclear envelope, allow substances that are produced in the nucleus to leave the nucleus and go out into the cytoplasm. And even things that are in the cytoplasm, if they're allowed to go into the nucleus. And then you see the term chromatin in your outline. So chromatin, and let me just say this, that this semester you're going to learn multiple terms for DNA, deoxyribonucleic acid. So DNA is found in different states, and it's still the same sequence of A's, C's, T's, and G's, but you find it in different forms, different states. Kind of a simple way to think of it is the different states you find water in, even though that's temperature dependent, and talking about DNA, it's not temperature dependent. But water, for example, is still the same chemical formula, H2O, but you could find it in a liquid, which we call water, a solid we call ice, and a gas, which is water vapor, but it's still H2O. So kind of the same loose principle then with DNA, that you find DNA in different states depending on what part of the cell cycle you are in. So here in this picture, you're looking at DNA in the state of chromatin. Chromatin is uncondensed DNA plus its proteins. So it's kind of long, stretched out. You can't count one individual piece of chromatin from another. It's really hard to. It would be almost like taking a ball of yarn and you just unwind it and you have this big, huge mess, and you can't see where if it's more than one piece, if it's 30 pieces, where one piece ends, one piece begins and ends. You can't tell. So that's DNA in the state of chromatin. We call that uncondensed DNA. In this class, you're going to learn about 6 different terms for DNA. That's your first one, chromatin. All right, so the main function of the nucleus is to house the DNA. And again, here it's in the state of chromatin. Now, one other structure here within the nucleus, this dark spot here, and here in the illustrations, this is an actual photograph on the left here. And then on the right is an illustration, but still drawn in there, the nucleolus. The nucleolus is where ribosomes are made in the nucleus. So these little round structures that are attached to the outside of the nuclear envelope and then the endoplasmic reticulum, those ribosomes are made in the nucleolus of the nucleus. Ribosomes are really important in protein synthesis. We'll see that when we get to chapter 10. Also, just so you know, sometimes, and it's really obvious in this photograph here, but sometimes the nucleolus is referred to as a dark body, and you can see why, because it's a dark spot within the nucleus, and it appears that way, I mean, especially when you're looking through a microscope, for example. All right. Now, the next thing in the outline is a category of organelles that I titled membranous canals and vacuoles. Notice I have a list there for you. It says includes the endoplasmic reticulum, the Golgi apparatus, lysosomes, and vacuoles. Then I typed out that brief sentence there for you. It says they work together to produce, modify, transport, store, secrete, and or digest macromolecules. So that might just be one short sentence, but it's meant to convey an enormous amount of weight to you, of high significance for this category. Because again, if we look at that sentence, it says they work together, they meaning this list of organelles, to do all these things to the macromolecules. Well, the macromolecules are carbohydrates, proteins, lipids, and nucleic acids. So this group of organelles is critical to the macromolecules that are in our cells. Very important group of organelles. So at the top of the next page, you see endoplasmic reticulum. So the endoplasmic reticulum is in two sections. So you have one that we refer to as the rough ER here, and then the smooth ER out to the outer edge. And notice that the rough ER section is contiguous or adjacent to the nuclear envelope. So how do you delineate rough and smooth ER? Well, it's the presence or absence of ribosomes. So the rough ER has these ribosomes attached to it. The smooth ER does not. So that's how we tell the difference. And location. So rough ER, again, is right up against the nuclear envelope, where the smooth ER is to the outer edge. As far as function of each, so the rough ER, because it has ribosomes, it is instrumental in protein synthesis. You cannot make proteins without ribosomes. So the rough ER makes ribosomes. The smooth ER, because it lacks ribosomes here, the smooth ER is the site of lipid synthesis. So it is the site of lipid synthesis. So it makes lipids. Now, the rough ER, let me just say another word about its location. There are chemicals that leave the nucleus and have to go into the rough ER to tell the endoplasmic reticulum what kind of protein to make. So things like messenger RNA. Messenger RNA is made in the nucleus. We'll see that when we get to chapter 10. That RNA leaves the nucleus and goes out to the rough ER. So the way that the endoplasmic reticulum has evolved, it's evolved to be right around the nucleus so that those chemicals don't have to travel out into the cytoplasm and risk potential damage by other chemicals before they convey that message on what proteins to make to the rough ER. So that's an evolutionary adaptation, the location of the rough ER. So that's pretty cool. So another interesting thing about, and here, this is just showing you that messenger RNA, the pink strand, interacting with the ribosome to make a protein. I'm going to teach you about this in chapter 10. But here, this is an important point to make to you. So the endoplasmic reticulum is a series of canals, membranous, it's made of membranes, plasma membrane, but it's a series of canals. And here's a ribosome, so this is your rough ER because there's a ribosome attached. When the products are made in the rough ER, so proteins, and then in the smooth ER, lipids, those products, like this glycoprotein here, have to be transported to their next destination within the cell. So a piece of the endoplasmic reticulum is literally pinched off around that product, like a little balloon, a little plasma membrane sac. So here you can see we're referring to it as a transport vesicle, but literally all it is a plasma membrane sac that was pinched off of the rough ER around that protein. That transport vesicle now is going to take that protein to its next destination. The smooth ER does the same thing with lipids. It'll pinch a piece of itself off around the endoplasmic, or that product from the smooth ER, that lipid, and send it off in a transport vesicle to its next destination. So please make sure that you read about both in your textbook and fill in your notes about the rough and smooth endoplasmic reticulum. It's a really important organelle because it makes proteins and it makes lipids. Those transport vesicles then leave the endoplasmic reticulum and they travel to the next organelle, which is the Golgi apparatus, also sometimes referred to as the Golgi complex. The second word really doesn't matter all that much. It's the first word that really matters. So you can see in your outline that I have 3 functions that the Golgi carry out. I have a 1, 2, and a 3. So number 1, it separates proteins and lipids that are received from the endoplasmic reticulum according to their destinations. So it tells the difference between a protein or lipid that's going to leave the cell versus one that will stay in the cell. Number 2, it modifies some molecules. And number 3, it repackages those chemicals into vesicles and sends them off out into the cell. So in the illustration here, you can see that the Golgi apparatus, it's like a stack of pancakes. So it's a series of flattened vacuoles. And we kind of think of the Golgi as having a receiving side and a shipping side. So the vesicles from the endoplasmic reticulum come in one end, and then they leave the opposite end, which is the shipping side of the Golgi. So take a look at the Golgi apparatus in your outline book or in your chapter in your textbook and fill in your outline. Now, the next thing to talk about in your outline are vesicles and vacuoles. So vacuoles and vesicles are similar to each other in that they are plasma membrane sacs. The difference is that a vacuole is much larger than a vesicle. Vacuoles carry large things, vesicles carrying small things. Now, while there are size ranges for them, I don't make you guys memorize those for this class. I just want you to know that lysosomes are larger than vesicles relative to each other. Here you're looking at like a food vacuole. Here's a vacuole for water to get rid of excess water in a protozoan. Plants have large, what we call central vacuoles, which are big plasma membrane sacs referred to as a vacuole to store water. So read about vesicles and vacuoles in your textbook to complete your notes. Then the last one in this category are lysosomes. So if I go back to this illustration here, so lysosomes carry hydrolytic enzyme, and enzymes are proteins. So on the slide here, you can see where it's labeled digestive enzyme. It's the same thing as hydrolytic. Hydrolytic means digestive, like hydrolysis reaction that I taught you in chapter 3. So lysosomes are kind of like the janitor of the cell. They will keep the cell clean of defective organelles, but then they're also involved in digestion. So this slide is looking at digestion, where you've got a food vacuole, so you have this large protein or carbohydrate being brought into a cell. The lysosome will fuse with it, and then that hydrolytic enzyme will digest that food. In this case, you see here some damaged organelles where the lysosome will fuse with a vacuole with a or vesicle with a organelle in it, and then digest that organelle. And that keeps the cytoplasm clean of defective organelles, because if your cytoplasm is cluttered with defective organelles, that could really impact the ability of the chemicals in the cell to move around and get from one place to another. So this slide's kind of nice. It's kind of orienting you with all of those things we just talked about. So you've got your nucleus, your rough ER, your smooth ER to the outer edge, vesicles that are leaving the ER, traveling to the Golgi, and then you have vesicles that are going to stay inside the cell, and then those that leave the cell. So and through your Golgi, you make lysosomes that the protein comes from the ER, rough ER, but then it travels to the Golgi and it's modified and repackaged, and it's a lysosome then with hydrolytic enzyme. So that brings us to the end of the first, or that large category of organelles. So again, please use your textbook to complete your notes as I kind of am going through the highlights of these organelles with you. And then energy related organelles. Excuse me. So energy-related organelles, here we're looking at really two in your outline under this category. So I've got the mitochondria on one page and then the chloroplast on another page in your outline. So let's talk about the mitochondria first. And that's the slide we're looking at here. So the mitochondria is considered to be the powerhouse of the cell. It is your organelle that makes cellular energy, which is ATP, adenosine triphosphate, is made in the mitochondria. But structurally, let's talk about that for a moment. So the structure of the mitochondria, it is a double membrane bound organelle. So it has an outer membrane and an inner plasma membrane. So these are plasma membranes. It has two, an outer plasma membrane and an inner one. So in your outline, you see the term cristae. The cristae are the inner membrane folded into these finger-like projections. They protrude into the middle of the mitochondria. So those are called cristae. And then the actual middle of the mitochondria is called the matrix. And that's a fluid-filled space. And these cristae project into the matrix of the mitochondria. Then I point out to you too, the space between the outer and inner membrane. It has a name, the inner membrane space. It's not very clever, but it works. And so kind of easy to remember because of that, I think. It's the inner membrane space. That location is actually going to be important in chapter 6. And so to mention this to you, in chapter 4 here, I'm teaching you about the different organelles. This organelle here, the mitochondria, is going to be the focus of chapter 6 when we get there. So in chapter 6, as I'm lecturing on that, in that particular lecture video, I will expect you to remember this anatomy for the mitochondria. I won't be reteaching it. I'm going to be applying that anatomy to the chemical reaction of cellular respiration. So you're going to want to make sure that you remember and learn the anatomy of the mitochondria. So in your outline, it says function. So the function is that it makes adenosine triphosphate. It makes ATP. That is its function, to make ATP. And so it says there, mitochondria carry on aerobic cellular respiration. That is true. Aerobic means with oxygen. So aerobic cellular respiration is the process in which plants and animals take glucose and we convert it to ATP, which is cellular energy. So again, that is something I will teach you about in chapter 6. We will learn about aerobic cellular respiration there. And then formula, there I'm talking about the chemical formula for aerobic cellular respiration. So I will recite the equation here. I don't have it on a slide in this presentation, but you can go into either chapter 4 or go to chapter 6 and look at the equation for aerobic cellular respiration there. But in general, this is, we'll start with glucose, which is C6H12O6, plus 6 oxygen. Atmospheric oxygen is O2. So plus 6 O2 yields, which is the arrow sign pointing to the right, 6 CO2 plus 6 H2O plus energy. And we'll leave it at that. So again, you can go to the chapters in the book 4 or 6 to take a look at the equation. Now let's go down to the chloroplast, which is on the next page. And then I have a general comment about both of them. So chloroplast, the chloroplast is again also an energy-related organelle, and it is found in plants and photosynthetic protozoans. It also has a double membrane. It has an outer membrane and an inner membrane. on the inside, in your outline, you see the term thylakoids. The thylakoids are these little flattened sacs here. They kind of look like those candy sprees that are stacked on top of each other. But those thylakoids are derived from the inner membrane. So the inner membrane gives rise to the thylakoids. And they're little flattened sacs, and they're actually connected on the inside. These stacks are connected. And then each stack with tubes of membrane, plasma membrane, are connected to each other. So all of these thylakoids are connected to each other. Now, in your outline, you also see the word granum. One stack of thylakoid by itself, this one stack, is a granum. Um on the end of a word is singular. If I wanted to talk about all of the stacks of thylakoids, I would put an A on the end, and it'd be grana, G-R-A-N-A. Grana is plural. So all of the stacks of thylakoids are grana together collectively. And then stroma. Stroma is the fluid-filled space that bathes the grana. So I'm overemphasizing, but to make sure you get the right word. The main function of the chloroplast is that it is the primary photosynthetic organelle. This is where photosynthesis occurs in eukaryotes. So it is the primary photosynthetic organelle. And in your outline, you see the term formula. So here, again, I can recite the equation for you, but if you want to see it clearer or in front of you on a piece of paper, you can go to chapter 7 and look at the equation for photosynthesis there. But it's sunlight plus 6 CO2 plus 6 H2O yields, which is the arrow to the right, C6H12O6, which is glucose, plus 6O2, which is atmospheric oxygen. So that is the equation for photosynthesis. And then the term chlorophyll, there is a green pigment. Chlorophyll is a protein, and it's the primary photosynthetic protein, chlorophyll. And so we'll talk about photosynthetic pigments when we get to chapter 7, because this organelle is going to be the focus of chapter 7. So as I told you with the mitochondria, that I'm going to expect you to remember the anatomy of the mitochondria for chapter 6. Same thing here. When we get to chapter 7 and we start talking about the actual chemical reaction of photosynthesis, I'll expect you to remember the anatomy of the chloroplast when we get to chapter 7, as we apply it to the photosynthetic reaction. Now, before we go on from the two energy-related organelles, I just wanted to mention a couple things to you, which is kind of cool. So when I guess let me do this. So when we talk about the mitochondria has a double plasma membrane, and so do chloroplasts. They have a double plasma membrane. Not only that, both of those two organelles, and it's not drawn in either illustration. You can't see it in here. But both of these organelles, the chloroplast and the mitochondria, have their own DNA. They have their own circular loop of DNA like bacteria. And that's because the mitochondria was once a free-living aerobic organism on its own. And the chloroplast was once a free-living bacteria called cyanobacteria, C-Y-A-N-O, that's the prefix, cyanobacteria, which still exist on the planet today, by the way. So mitochondria and chloroplasts replicate themselves. They're not replicated or created inside of a cell by the endoplasmic reticulum and the Golgi and so forth. They're self-replicating because, again, they were once free-living bacteria. Aerobic bacteria for mitochondria and cyanobacteria for the chloroplast. So what you're looking at here is looking at events where the endoplasm or the mitochondria was engulfed into cells, and so were the chloroplasts. So if we go back to our generic plant and animal cell, if you look at the animal cell, so animal cells have mitochondria, and so do plant cells. So both plants and animals have mitochondria. So do fungus, so do protozoans. That means that the incorporation of mitochondria into eukaryotic cells happened before the split of plants and animals and fungus from each other in a common ancestor. So you and I have mitochondria in all of your cells that are carrying out aerobic cellular respiration for you. So you and I all have these bacteria, essentially, these highly evolved bacteria that live in our cells that help us make ATP, cellular energy. Whereas with the plant cell, if you look at the chloroplast, so plant cells have not only mitochondria, but they also have those chloroplasts to make their own food. If you go back to the animal cell, which I know you already knew this, we don't have chloroplasts. We don't make our own food. We can't. Instead, we have to ingest other plants and animals to gain our energy and nutrition. So chloroplasts were incorporated into plant cells after the split between plants and animals and fungus. But plants then have both types of highly evolved bacteria that live in their cells. So they've got both mitochondria and chloroplasts. So essentially, plants make sugar in the chloroplasts, then they move it into the mitochondria to make ATP. You and I have to eat our food, plant and animal, and then we take that sugar that we've ingested, and that's used in the mitochondria to make ATP for us. So pretty cool stuff. And that brings us to the last section in this chapter, which is the cytoskeleton. So the cytoskeleton is part of the cytoplasm. So the cytoplasm, as I kind of told you at the beginning of this chapter 4, is that it is a water-based environment with salts and organic molecules in it, which is still true, but there's other things in it. And so the cytoplasm is kind of like a hair gel consistency. It's not as fluid as water coming out of the tap in your kitchen faucet. It's thicker than that, so it's more viscous. So again, I think like hair gel is kind of a simple way to think of it. But throughout that gel-like cytoplasm, there is structure to it, and that's what the cytoskeleton is. The cytoskeleton is composed of these different fibers that are made of protein. So these different protein fibers that are of different diameters. So the really small fibers are called microfilaments. The medium-sized ones are intermediate. The larger ones are microtubules. And you'll notice then in your outline, I have 4 functions of the cytoskeleton. Number 1 is they help maintain cell shape. I want you to read about each of these functions in your textbook and fill in more detail about them. Number 2 is cell movement. So they help cells move, especially the microfilaments here. The third one, function, is organelle movement. So organelles, like vesicles and vacuoles, move along these fibers, especially the intermediate fibers, in order to get from one point of the cell to another. That way they're just not spinning in that cytoplasm material. And then the fourth function is cell division. And here the microtubules are most involved in cell division, the microtubules. So I'm not going to say much more about cell division here other than those microtubules are going to attach to the DNA and help the DNA move around through cell division. But I'm going to teach you cell division in chapter 8. So I think I'll just leave it at that. Now, in this slide, you can see that, and in your textbook, it gives you diameters. I'm not going to ask you to memorize the numbers, but I do want you to know that microfilaments are the smallest in diameter, intermediate, medium. Microtubules are the largest in diameter as far as size. So go back into your textbook and read about these different fibers and the 4 functions of the cytoskeleton and fill in your notes. So you have detailed notes there to study for the exam. And then cilia and flagellum, which is the last page of the outline. So cilia and flagellum are both composed of microtubules. So here is a cross section of a microtubule. So they both have a 9+2 microtubule arrangement. And in this cross section here, you can see that arrangement. So the 9 is 9 pairs of microtubules around the outside. So if I count starting here, 1, 2, 3, 4, 5, 6, 7, 8, 9. Then 2 individual microtubules in the middle. So 9 pairs around the outside, 2 individual ones in the middle. 9+2 microtubule arrangement. The other important thing to tell you about cilia and flagellum is that they are both extensions of the plasma membrane. So you can see the plasma membrane here. This is the microtubule arrangement in the middle. So the plasma membrane, these are extensions of the plasma membrane. The plasma membrane covers them. So look in your textbook to help you distinguish between cilia and flagellum, their functions and how to tell them apart. And then I have a couple pictures in here. So here is cilia. Where there's one, there's many on a cell. So this is from the human respiratory tract. And then here is a flagellum. Flagellum are more hair-like, whip-like. They're longer. And both are motility structures and so forth. But take a look at cilia and flagellum in your textbook to fill in your notes there. And that brings us to the end of Chapter 4 lecture video.